For the opportunity to give a talk here today. It's been a really, really interesting conference so far, and I look forward to the rest of the day. I'm going to talk to you about a piece of our current work, which is aimed at understanding the causes and consequences and potential evolutionary significance of early life effects on later life traits. This work is, of course, strongly motivated by work on early life effects on health and disease in humans. You guys have already seen, I think, probably both of these examples in the context of this conference. But just to recap, they are some particularly striking examples of early life effects whereby individuals who were exposed to severe deprivation during a sensitive developmental period exhibited higher rates, higher susceptibility to a variety of diseases, including cardiovascular disease, metabolic syndrome, and schizophrenia later in life. This kind of effect is not confined to humans. So here's 3 examples from a single paper that focused on relatively long-lived ungulate populations. So in each of these 3 examples, the authors showed evidence that early life adversity predicted overall lifespan. So in mountain goats, do I have a, nope. So in mountain goats, each of these dots, I should point out, is actually a birth cohort, not an individual. So there's a lot more data here that actually looks like there is based on the scatter plots. So in mountain goats, individuals who were born during years where spring came earlier and there was more available vegetation lived longer. In bighorn sheep, individuals born in birth cohorts where there was less resource competition due to population density lived longer. In roe deer, individuals who were born in years where there was adequate rainfall tended to enjoy a longer lifespan. Okay? So the cumulative evidence across both humans and non-human animals suggests that early life experiences can have some fairly potent effects on traits that we can very readily imagine to be adaptively important. And so by studying non-human animals, we think we may be able to get a window into how these effects might work and what their evolutionary significance might be. So the population that I'm going to talk about today is a wild population of baboons, yellow baboons, Papiocynocephalus, residing in an arid savanna-like environment in East Africa. So this is in the Amboseli ecosystem of Kenya. I'm very fortunate to work on this population. This is a population that has been subject to longitudinal study on a near daily basis since 1971. The study was founded by Jean and Stuart Altman, who are at Princeton, and is currently co-directed by Jean, Susan Alberts, who's at Duke, Beth Archie at University of Notre Dame, and myself. Okay, so what we're trying to do here is distinguish between 2 hypotheses for the evolution of early life effects. Oops. That was really important. I want to point out that this work is really spearheaded by Amanda Leah, who's a graduate student co-advised by Susan and myself and is actually here. So I'm sure would be happy to answer questions about this work as well. So what's our goal here? We're trying to distinguish between 2 alternative hypotheses for the observation of early life effects on later life traits. One is a hypothesis that was very well covered this morning. This is an early life programming or a predictive adaptive response hypothesis. And if we were to write this down in the context of reaction norms, like I've done here, what it's saying, we think, is that individuals in green, individuals born in adverse early conditions, will tend to have lower overall fitness, shown on the y-axis, if they experience mismatched environments in adulthood, right? Good adult environments. In contrast, if they experience matched environments in adulthood, so poor quality adult environments, in addition to early adversity, they should actually experience a fitness benefit. So you get these crossing reaction norms. An alternative hypothesis is one of pure developmental constraints, okay? And so in this case, it's very simple. Individuals who experience early adversity are at a disadvantage, regardless of the quality of their adult environment. These 2 hypotheses can be quite difficult to differentiate from one another, particularly because often we have data for individuals who are born in poor early environments and born in good early environments, but only in cases where they experience one particular environment in adulthood, particularly high-quality environments. And so what we're essentially doing is sampling those 2 points that I've circled in red. Okay? And if those are the data that you have, you don't actually know what the rest of the reaction norm looks like. And so it could be a crossing reaction norm consistent with the hypothesis on the left, or the reaction norms could, in fact, look something, oops, like this, right? And that's much more consistent with a developmental constraints hypothesis. So we're arguing that you need to be able to identify the fitness consequences of early adversity versus high quality early conditions in both high quality conditions and adverse conditions later on in life to tell the difference between these two hypotheses. Okay, so this is facilitated if you happen to study a system like I do where there's a lot of environmental variation. So what I'm showing you here is cumulative yearly rainfall on the y-axis by hydrological year in Ambicelli running back until the mid-70s. Sometimes it looks like that flooded landscape on the left, and sometimes it looks like that dry, arid savanna on the right-hand side. And just to give you an idea of where we're talking about in rainfall space, this is where Tempe falls. So it's sometimes drier than the sort of desert-like conditions here in Arizona. Okay, so we were particularly interested in what happened to females in our population during this year, 2009. That was the worst drought on record ever recorded for Ambicelli, and we saw huge amounts of ungulate die-off in particular during this year. So the fact that it was so bad was really compounded by the fact that, as you can see, the year before, 2008, was also a particularly dry year. So here we have substantial ecological variability from year to year. As a fitness-related trait, I'm going to talk about fertility-related traits, and particularly both the ability to resume estrus cycling after postpartum amenorrhea and conception rates. So here I'm plotting the same kind of x-axis is on the left-hand side. On the right-hand side, this is hydrological year, but the y-axis has been swapped out for conception rate. So this is conceptions observed per adult female in the population per year. And you can see that there's some variation, but it tends to hover around 65% or so. In 2009, that took a precipitous drop. We didn't see a lot of elevated mortality, but we saw about a 25% decrease in conception rates for females in the population. So something that we feel is arguably a pretty good fitness-related trait. Okay, so this allows us to test these two hypotheses by looking at a poor quality adult environment, in this case during the 2009 drought, and contrasting it to high quality or moderate environments that saw neither excessive rainfall nor severe drought. And to contrast the effects of those two different environments within individuals, so essentially we're plotting a reaction norm for every individual here, for individuals who were born in very dry environments versus those that were born in what we're defining as moderate or high quality environments. And I just want to point out that our results are really robust to how we define what a moderate or high quality or a poor early environment is, but I won't be able to talk about that because of time constraints. So here's the question. How does the shift between a good year and a poor year affect females who were born in good years as opposed to females who were born in poor years? And because reproductive capacity is affected by a lot of different things, in all our models we control for things like dominance rank of the female, her reproductive status, the size of her group, indexing resource competition, and also various aspects of her age. Okay, so do we see early life effects? We do. And does that differ between females who were born in low-quality years on the left and females who were born in high-quality years on the right? So I'm just going to walk you through this results picture. On the y-axis, we have a measure of essentially the likelihood of conception from the models that we fit. So higher is more likely to conceive, lower is less likely to conceive, and each of those sideways plots gives you a sense of the distribution for females in a particular category. So what we're seeing here is that for females who are born in low-quality years on the left, there's a bigger difference between their within individual likelihood of conception in high quality years versus in the 2009 drought as compared to females who are born in low quality years. We actually see the same qualitative results whether our response variable is conception probability or the likelihood that she resumes estrus cycling after postpartum amenorrhea. So to summarize what those results look like, essentially we are seeing no evidence that females who were born in drought actually do better in matched environments, in drought environments in adulthood. In fact, we see the opposite effect. Females who were born during drought conditions do worse during drought conditions they experience in adulthood. Okay? In contrast, females who are born during these moderate or high quality years actually do a little bit of a better job buffering themselves against these costly environments in adulthood. Okay, so again, I want to stress that this particular comparison is a within female comparison. How does a particular female do in good years versus bad years, depending on her early environment? One can also ask the question, well then, do you see differences between individuals born in good years and adverse years in the 2009 drought itself, so during those costly years. And in fact, we see maybe a trend towards significance here, but it's not significant. However, the direction of the effect is that females born in low-quality environments do a little bit worse. What explains the difference between the within individual and the between group comparisons? One possibility is heterogeneity imposed by other aspects of the environment. And this appears to be part of what we're seeing. So here, I'm showing you only the females who were born during a drought, who also lived during this particularly severe drought in 2009. And what I'm showing you is a measure, again, of how well they do in terms of their ability to resume cycling or their ability to conceive, with the x-axis now being their social status at birth, the dominance rank of their mothers when they were born. And what we see in both cases, for cycling probabilities on the right and conception probabilities on the left, is that high-ranking daughters of high-ranking females, tend to also be fairly buffered from the effects of drought conditions, whereas daughters of low-ranking females take the biggest hit overall to fertility. So I think all this is doing is really emphasizing that early adversity is multidimensional, that there are many, many things that can happen in early life besides simply resource deprivation. Okay, so together we think we're seeing evidence primarily for contingently experienced developmental constraints and not adaptive programming, where the contingency works in two ways. One way is dependent on the adult environment. So we see these differences between the two groups of females when the adult environment is poor. We actually see no difference between their performance with regards to these fertility-related measures when the environment is good. The other contingency that we're talking about here is the one imposed by the social environment. So poor quality environmental effects appear to be buffered out, to some degree at least, by high quality early social environments. So does this matter? Well, females who are born in poor environments took a really, really big nosedive during 2009, a 60% decrease in conception probabilities. That was only a 10% decrease for females who were born in high-quality environments. For those females who were born in low-quality years and lived through low-quality years, our model predicts almost a 2% increase in conception probabilities every increase in maternal rank position. Okay? So that seems small on the face of it, but some of these groups can actually include dozens of females. So that buffering effect could be actually pretty substantial if we compare the highest-ranking and lowest-ranking females in a group. Okay, so I think together, our data set, at least with respect to these traits, supports a developmental constraints model much more so than it supports a predictive adaptive response model. And it actually joins with data that's been published recently on roe deer and on fins in humans, suggesting that developmental constraints is a better explanation than predictive adaptive responses. Now, there are data that support predictive adaptive response models, and some of the best examples come from species like Daphnia or locusts or voles. And one of the things that might be really striking to you as a difference between the species on the left-hand side and the right-hand side is simply lifespan. Okay? And I think that we don't want to toss out adaptation altogether, but it's useful to think about the cases in which predictive adaptive responses should emerge. This is not our work. This is work by Dustin Rubenstein and Carlos Botero, just published this year in PNAS, which posits circumstances in which irreversible plasticity, the thing that I think is closest to predictive adaptive response models, should evolve. And where should it evolve? In cases where environmental cycles are longer than generation times, and in cases when the environment is actually fairly predictable from early environments. Where do baboons live, and where do I think humans live? Down here. Okay? So that's actually going to disfavor, I think, an adaptive explanation for the sorts of plasticity effects that we tend to see. Okay? And I just want to wrap up and thank my co-authors and collaborators and the folks who contributed to this work.